Okay, welcome everybody. We'll get going with this. I know you all remember the test. First exam opens today at 10. It's open through the weekend, closes Monday at 5 p.m. There are 30 questions, most multiple choice. There's a couple of matching and a little bit of true false. It should be smooth sailing, I would think. The test is set to be timed for 2 hours. I don't think it's going to take 2 hours, but sometimes open book and open note exams may take a little longer. So there you go. Questions or concerns about that? Okay, so we left off a chunk of the way into this chapter, and it looks like there's a whole bunch of material left, but really, I think we can move through here relatively quickly and hit the key bits, right? As I mentioned, as we were getting into some of this chapter, a good chunk, I feel like, is some review. When we start thinking about some of the cellular details, and some of it really focusing in on the bacteria might be, probably would be new material. All right. So when we think about structures of bacteria, we've mentioned a little bit about using some staining techniques to give us some ideas about types of bacteria. And the gram stain, by looking at components of the cell wall, thick or thin, gives us various reactions in the gram stain, and we call them gram positive or gram negative cells based off of the thickness of that. And we'll see some images of cell wall stuff in a second, all right? We can also see some morphology. Are they rod or bacillus shaped, or are they a coccus or cocci shaped? Giving us some clues about the organisms based off of that. All right. With that said, there's potentials for some variation. Kind of depends on the types of microbes we're talking about, where they might be surviving, as far as some of these features that they may have. All right. And in some circumstances, we think of it as maybe from the disease standpoint, increasing the chances that maybe they could get in, take hold, and maybe also be undetected by our own immune responses to actually establish themselves and start causing disease. So there are some potential maybe advantages for these bacteria to have some of these things, or from our standpoint, maybe disadvantages, right? Because it gives them a leg up. So typically when we think about our prokaryotes, they've got a cell membrane. Their chromosomal DNA is circular, a single chromosome, and it's oftentimes described as being in a region called the nucleoid, but it's not a membrane-bound vesicle. It's not an enclosed, encapsulated structure. It's just kind of in the middle of the cell, all right? Technically, there are some features when we think a little bit about some of the genetics that we'll talk about as far as helping the DNA get partitioned through or to opposite ends as it's trying to replicate, but we're not going to focus on that right now. Sometimes bacteria can harbor some extra chromosomal material. We call that stuff plasmids, and it's this littler rubber band looking structure down below. These things, chromosomal material, are in some bacteria. They can live without them, but they live better typically if they have it, giving them opportunities to survive on different substances or to potentially be resistant to numerous types of antibiotics. And in some circumstances, having some of these plasmids allows that bacteria the opportunity to share it with others. Conjugation is the transfer of typically plasmid material from one bacterium to another, all right? We'll talk about some of that when we think some biotechnology stuff, all right? So They may have this outer bit that's a sugary coating, this glycocalyx, or capsule layer. And then as you know, we peel it back, the cell wall and the membrane, and we move into the cytoplasm with all sorts of little speckles, the ribosomes, where we think about protein synthesis. And different than our eukaryotic cells, where we have maybe two different locations where this can be happening, rough endoplasmic reticula and in the cytoplasm. In bacteria, it's just the cytoplasm, right? So there's potentially some inclusion bodies inside of these cells that we might see, maybe some food molecules or other substances that they may be harboring or holding inside of them. Iron deposits could be in some bacteria that in turn might be living maybe deep in the ocean or something else like that. So some of them have flagella. Here is a single flagellum, plural, we would say flagella, and it might be a single flagella at one end. We call that monotrichous. It could be completely covered all over with flagella. That's paratrichous. It could be a tuft at one end, lopotrichous, and at either end, a flagellum, amphitrichous. All right, handful of different names, but it's just a way of describing the way their flagella appear. All right, we'll show it in a couple of slides a little bit about it as far as how the flagella functions in bacteria. There's similarities between eukaryotes that have flagella, but there's definitely some pretty big differences as far as structure. I mean, the end goal is really movement, but the way they go about it is different, right? So some of them also have pili. This pilus right here could be something coded for by a plasmid that allows one cell to make contact or communicate with another. All right. The fimbriae are little shorter hair-like projections, not the same, but sort of like cilia when we think about some eukaryotic cells. All right. And the fimbriae, I think of them in bacteria kind of like a Velcro, allowing cells to stick to other stuff, to maybe take hold for attachment. All right. And in those circumstances, might help with disease progression. All right. So handfuls of different possibilities. And when we look at them, sizes and shapes are varied. All right. They are all microscopic, and we've got some electron micrographs on the side to give us maybe a little more detail. All right. So coccus, as I said, or cocci, if we're saying many of them, all right, are spherical shaped, right? And as far as, not flipping it back, but as far as the flagella go, really it's the bacillus or the rod-shaped Vibrio as well that have flagella. The cocci, the spherical ones, don't have that for their movement, right? And spirochetes, they're a little bit different with how the flagella is kind of twisted around their body to make them corkscrewed-like, right? So don't really see it, but it's there as like these axial filaments. So bacillus or bacilli, these are rod-shaped, and we oftentimes interchange rods and bacillus. There is a genus Bacillus as well, and they all look like this, right? Rod-shaped, right? Maybe various sizes, but like that. A little bit of a curved rod, we call those the vibrios, and they're sort of comma-shaped, right? Sometimes, you know, it's difficult to really say, I mean, that looks sort of curved just like that, right? Is that a vibrio or just a bacillus, right? Sometimes you get a little bit of one end that's a little bit wider than the other, so it looks a little more comma shaped, and that would you maybe say with a little more certainty that that's what's up, right? The coccobacillus, these are sort of shortened rods or elongated cocci. It's in between, right? Kind of odd, right? And, you know, I sort of think of them as it's sort of a stumpy bacillus, but sort of egg-shaped, right? And then the spirillum and the spirochetes, these are the curved, but more curved than the vibrio, spiral shaped, but it might be loose like the spirillum or really tight twisting the spirochetes. And you can see some definite differences with the curving of that that allows us to think about it. So looking at the organisms under a microscope, we can see the size, the shapes like we just saw. We can see their sizes that are all relatively about the same, all right? There are some rods or bacillus-shaped ones that are really pretty small. There are other ones that are almost huge compared to them, all right? Oftentimes the gram-positive bacillus are a bit bigger looking than the gram-negatives when we think about those, all right? With the others, like the coccus-shaped bacteria, a lot of these on here are more really what we think about looking through a microscope and thinking about cocci bacteria, all right? The bacillus bacteria, typically you see rod-shaped bacteria spread out all over, sometimes linked up, but that's not something that really lends to anything that's super informative, all right? So they might be streptobacillus or chained. Sometimes we call that cords, but like I said, not something that really clues me in like, oh, we're talking about these specific types of bacteria, all right? It's just maybe a feature of how they've been replicating and you're able to kind of catch a glimpse of that, right? But the spherical shaped or the cocci shaped bacteria, there's all sorts of potential variation with them. Sometimes you see them as singlets, little spheres all over on the slide, and they look pretty well kind of spread out and just little balls all over on there, all right? Purple or pink, gram-positive or negative, but separate, all right? And we would say, you know, like I said, an individual coccus or many cocci on that slide. Then you might see other opportunities where instead of them just being singlets, many of them are just linked up as doublets. We call that diplococci. And that might be kind of informative when we're looking at a few of these, like Streptococcus pneumoniae. It often looks like this, all right? Some of them get groups of fours, a tetrad formation. There are others, and we're not seeing it right here, but they compound on themselves and give you more kind of a more three-dimensional, cube-shaped. We'll move through and think a little of that later, but we call that Sarcinia as they stack up. The diplos can continue to elongate, really, as we see them linked like that. And we think of this as a streptococcus, a chain type of shape. And they can be pretty long when we think about it through a microscope, looking at them through a microscope, numerous ones all linked together. And they typically look very different than the ones below that, the streptococci that are clustered, right? So the way these cells are dividing, when we get the chains, they're basically just splitting in half and splitting in half, and they keep linked together. But with the staphylococcus, it's irregular planes of division. It's not just splitting in half. We might cut the other way, or we cut at an angle, and then we get these clusters or groups of cells that are in all sorts of different angles poking out three-dimensionally, right? Grape-like, chain-like, stacked up, linked up, or singles, right? All sorts of different arrangements, and seeing it through the microscope may clue us into a little bit about what we're thinking about, right? And maybe based off of the way they appear and their gram reactions might tell us different types of auger that we might be able to use to grow them and to separate and maybe distinguish them a little better. And we're going to learn some of that stuff in the lab as we progress through here in the next few weeks, thinking about different types of media. So we want to think about how cells are pulling stuff in, and oftentimes diffusion is one of the main mechanisms. It doesn't matter if we're talking bacteria or eukaryotic cells, things being able to kind of readily move through the membrane is important for the acquisition of nutrients. And when it comes to bacteria, it's definitely one of their main features of pulling in nutrients. So this, hopefully some review stuff. In an isotonic solution, the concentrations, whether it's the same stuff out and in, doesn't matter. But if it's concentrations are the same, we say that it's an isotonic solution, and the net movement of water in or out of that cell is balanced, right? There's no directional flow, all right? And that's why you've got arrows in both directions. In a hypertonic solution, there's more dissolved solutes on the outside of the cell than on the inside. And so that inside of the cell, there's more water. And in turn, water wants to go out of the cell into the solution, and the cell shrivels up. We call that crenation. In a hypotonic, it's the opposite. And there's less solutes dissolved on the outside than the in, and water wants to go into those cells, causing them to swell up and to potentially lyse or to explode. Right? Bacteria, having a cell wall, gives them a bit of protection in some of these circumstances, but not 100%. Right? And in either circumstance, it could be bad news. Right? And when we think about food stuff, like preservation of stuff, this hypertonic situation becomes pretty important. There's lots of things we might utilize for preservation, like adding a boatload of sugar to some fruit and making jam or jelly, right? Or loads of salt and salting something out and causing it to be preserved that way. Meats maybe that way, or drying, which is also a similar sort of situation with fruits and jerky, things like that. Pickling is another situation where it's hypertonic, and it makes it so that those bacteria typically can't survive in those circumstances. There are some potentials, like maybe preserves, jams, or jellies, especially things that maybe you've produced at your house. Oftentimes things that you buy at a supermarket have some other things in there that are preservatives that are preventing things like molds from growing. But if you make it at your house with just some sugar and some fruits, probably a greater likelihood that that jelly in the fridge for extended periods of time might get a little bit of mold in there, because it can kind of tolerate these higher concentrations. But bacteria, that's really kind of what we're trying to prevent in the first place. Usually they don't tolerate these situations that we might throw at them. So this really, we're looking more at, as I see this, these are plant cells, all right? But we say right here prokaryotic cells, right? But these look like a bunch of plant cells to me. And part of that is just, you know, using figures interchangeably, because there is some similarity here, right? But this isn't really a prokaryotic cell. There is a humongous vacuole that we see inside of a plant. We're not seeing their little chloroplasts, but they're there, right? So plants and our bacteria deal with it a little bit differently. And the cell wall doesn't matter who we're really talking about. The components of it are different, but structural support, it's the same idea, all right? So the plasma membrane in a hypertonic solution, these cells lose a bunch of water and it shrivels up on the inside, but the outside, the shell of it still lets it kind of look as if it's the same size, all right? And in a hypotonic, where it pulls in extra water, in a plant, the vacuole picks up a lot of that, but it pushes the cell out. This plasma membrane gets pushed really, you know, tight against the wall, and it makes it kind of get a little plump, if you will, compared to the normal cell. It bulges out just the littlest bit, all right? But the cell wall helps to prevent it from lysing, from just exploding. If it's an animal cell in a hypotonic solution, it would explode, right? It would lyse. And if we're talking red blood cells, we call that hemolysis, the lysis of those red cells. So this image Electron micrograph, we've got a dash line going around this, looking at the DNA inside of, not inside, but inside of my green dash in this region that we call a nucleoid region. And you can see part of the chromosome a little bit darker. That's what we're seeing here with the ends, the nucleotides. So the speckles in that first image that we started with, the little ribosomes that are all over in the cytoplasm, are open and available to basically start building proteins as soon as we start making that message from the DNA, right? And in bacteria, it's virtually all business, all genes, right? All coding, right? We don't have any of the extra in-betweens that we see in your eukaryotic cells. So the instant, almost the instant that we start making messenger RNA, it can link up with ribosomes and we can start bringing in transfer RNAs and building proteins. We don't need to process, we don't need to do anything like that. It is almost the second we start saying, let's build a protein, we make it, right? And as we start moving through that message, we're still building more message. As those ribosomes are pulling through there, multiple ribosomes can be linking up to this message. And it's like a little assembly line, a little factory where we're pumping out massive amounts of proteins simultaneously. When we think about genetic engineering, utilizing bacteria for those purposes are truly awesome because we can have it happening very rapidly, right? We'll talk some details about how do we get eukaryotic information into prokaryotic cells, because we got to do a little bit of manipulating to get that stuff going on, all right? So there are other types of things we may see. We showed a little of that, these inclusions. So different things that cell may be harboring, some lipid droplets, different types of stain, these granules within there, that some pigments, sulfurs, I said some iron, these magnetosomes, and maybe some gas vacuoles with some others that we might think of lending to some buoyancy, somebody living out in a little pond. So we talked about the spore formers, the bacillus or the clostridium that can form spores and basically go on to live another day when conditions are a little unfavorable. I just looked and I'm like, let me set the mood. There, isn't that better? All right. So when a bacterium that's got the ability to form a spore, when something happens, it's too hot, it's too cold, I think I'm starving to death, right? You're looking at me like he's not starving, right? I'm not talking about myself, right? All sorts of chemical cues potentially that bacterium may be sensing in its surroundings, something that's like, holy crap, I'm gonna die, right? Causes it to go through this process, all right? It replicates its DNA, but it's not replicating it like it's ready for cell division, right? And bacteria divide through their process that we call binary fission. They copy and then they elongate and separate, right? So instead of doing that, it replicates it and it starts packaging it up and condensing it, getting it prepared to basically persist anything we think we could really throw at it, almost anything, all right? So membranes start to surround it, basically building a shell around it, more stuff deposited. It packages up some other stuff besides the chromosome in there. There's some enzymes needed to get it all back going and start the process of being a bacterium again. It's more than just that. It all gets packaged up together in this spore, right? A cortex and a harder, more impenetrable shell starts to form around this spore. And in the process of this, the bacteria that was, trying to survive really stops that. The process of it going on living really is done. And it starts to break down and the cell ruptures and opens up, releasing this spore to basically just hang out, right? And there are opportunities for these things to persist kind of indefinitely, right? There are mechanisms that can kill them, like, extreme heat, well above boiling temperatures with an autoclave, right? Or some irradiation, something like that, right? But just out in the soil or something like that, they could potentially persist indefinitely. They're not living, right? They're just waiting, right? And once the conditions become favorable, like stepping on a rusty nail and getting Clostridium spores into your foot, all of the sudden, conditions for that little spore are optimal and it starts to grow again, all right? Just depends, right? This, I told you some Bacillus and Clostridium. This is Carboxyandothermus hydrogenoformans. I don't even know if I said that right. Doesn't matter. These guys we don't care about in here, all right? There are a few other spore formers, all right? But when it comes to the real ones that are on our radar as potential disease or facelift givers with our Botox, right? These things are the ones that are important when we think about medical sorts of stuff. Not that these aren't important out in the environment, all right, but they aren't something that we think about as a human pathogen. So membranes, everything's got it that's living, right? Plasma membrane and bacterial membranes. There are some variations between them and eukaryotic membranes. There are technically a little bit of variation between bacteria and archaea when we think about their plasma membranes. And the archaea plasma membrane bears, it's kind of in the middle. It's got some things that are bacteria-like, and it's got some stuff in the plasma membrane that are eukaryotic-like. They're kind of in between, all right? They're still prokaryotic cells, but they've got some features that are sort of in betweens, if you will, all right? So features like, the phospholipid bilayer, common, right? Different structures like substances, sterols in the membrane, like cholesterols in our own, or some deviations when we think about some others. Sugars and proteins and lipids linked together on the surface, lending to a signal that says this is this type of bacterium, and our immune cells potentially seeking out some of that to recognize as being foreign, right? Opportunities for things to move into or out of the cell through the membrane through processes like diffusion or facilitated transport happen this way. So diffusion is truly a passive process where molecules are able to move down their concentration gradient. So an area where it's super concentrated to where it's not, and this is like time-lapse photography. So a bunch on the outside of the cell, hardly nothing on the inside, or nothing as it appears. And in turn, we would like to move through there, down our concentration gradient. Molecules that are able to readily move through the membrane can do this through simple diffusion. Others that can't simply go right through the membrane may still move by diffusion, but we call it facilitated, and it's a protein channel, a pore that's in the membrane that allows them to move down their concentration gradient. Or maybe it's something that doesn't work that way, and we have to actually force it through, maybe still through a channel, likely through a channel, but we use energy to do that, and we call that active transport. It's not a passive process, right? It's pushing things against their gradient. This one is just simple diffusion, moving down the concentration gradient. And over the course of time, we reach equilibrium. We're balanced, all right? Same amounts outside and in, all right? The facilitated using those channels. And with active transport, linking up something, ATP molecules linked to that channel. In this case, it's sodiums. The sodiums get pushed to the outside of the cell. And while it's still open, this protein channel, potassium ions are able to hitchhike a ride on that channel, and they get pushed to the inside. We shove 3 sodiums out and let 2 potassiums in. And as this happens, it creates a charge imbalance across the membrane of that cell. And that becomes super important when we think about all sorts of activities at a cellular level, whether we're talking bacteria or our own cells. This becomes important. So this image is of a cell wall of our bacteria, or cell walls, because we've got the positives and the negatives together. The cell wall material in bacteria is made up of a cross-linking of sugars and proteins, and we call it peptidoglycan, all right? The peptido is the protein and the glycan is the sugar, all right? And they are linked together. In gram-positives, they are like fused, one sheet on top of the other to get a very thick cell wall. And in a gram-negative, it's just a single sheet. They're not stacked up on one another, all right? So the little pentapeptides that we see in our gram-positives are what's giving us multiple stackings or layers of sheets together. It cross-links them together, all right? So both of them have the NAG and the NAM, N-acetylglucosamine and N-acetylmuramic acid. They are linked together to give us this cell wall material that makes it very, rigid, gives it its 3-dimensional shape and protection, right? It's similar like in plants, all right? But plants use different sugars. They use celluloses, linking them together or glucoses, excuse me, to make the celluloses. They link those glucoses together. These different sugar and protein cross-linkings, but it's a similar idea deep down. So in those gram-positives, there's a comparison between the positives and the negatives. There are variations with it as well. This blue stuff is the multiple layers of peptidoglycan. And again, those pentapeptides helping them to all kind of hold together into a big, thick blanket, if you will, versus a thin sheet over here, all right? There are other substances, some teichoic acids. Gram-positives have teichoic acids, gram-negatives don't. And with the gram-negative, they've got an outer membrane that we call a lipopolysaccharide layer, this pink bit right here, all right? Well, more than just the pink bit, this bit right above it, all right? And on it are a handful of different antigens. Lipid A and O antigen are 2 things that the gram-negatives have that can stimulate our body to say, these are bad news, and it gives us some ill effects. We'll touch on that later on, all right? There is a bit of space in between here, this periplasmic space in between this membrane. And as we move through and start thinking about metabolism, this becomes important because it's going to serve a similar role like what we think about the inner membrane space in mitochondria do, all right? So different, all right? There are some other features, as we described a little bit when we were thinking some staining stuff with those acid-fast bacteria. Like I said, they're technically gram-positives, but usually we don't know that because this mycolic acid, this waxy substance, basically coats the outer bit of that peptidoglycan and makes it so gram reactions, gram staining reagents don't penetrate through there, all right? We typically use some heat, a steam bath, to get those cell walls, to get the wax layer to really loosen up and allow stain to penetrate through. And without it, it just doesn't happen, all right? So the mycolic acid we see in these acid-fast bacteria, and it makes it so that gram stains don't work, all right? By heating it, it makes it more permeable, and the carbofuscin, which is the stain for acid-fast bacteria, is able to get in there and take hold. This is just a blow up of the outer membrane of those gram negatives. So this first bit of that, outer membrane, there's another bit of fatty acid down below that. We're not showing that, but it's right here making that outer membrane. And then the O antigen and the lipid A is this first little bit all together as one piece. Not going to go into great detail, but real quickly, when we think about some gram-negative infections, not all right, but E. coli is a pretty good one as an example. It can cause some bad GI issues, potentially fatal, all right? Depends on the strain of it, and it also depends on the patient. Really little kid or an elderly person are more susceptible to super ill effects with some of this bad E. coli, all right? Treating with antibiotics, we might think is a great idea for everything. Just try to kill these bacteria. But when it comes to some of these circumstances, like maybe E. coli, that might not be the best idea, because if we are killing the bacteria, we are potentially causing greater issues. We release a tremendous amount of these antigens, the lipid A and the O antigen. As that bacterium is being destroyed, it releases a boatload of this stuff, and that's what's causing some of the ill effects. Now you've compounded that, right? You've made the issue much worse, right? So sometimes we may treat, but other times we think more like fluid therapy, fluid replacement and electrolytes, but not trying to kill off stuff with antibiotics because it might compound the ill effects, all right? Depends, all right, on those circumstances. So here's an image, this bluish greenish bit, the glycocalyx is that sugary coating, and we can see some of that with some staining techniques. We can also potentially see some of it when it grows up on a petri dish, because the colonies oftentimes look very shiny or slimy, kind of mucoid sort of an appearance. That's pretty indicative of a sugar coating or a capsule layer that they're producing. And this bacterium Pseudomonas is pretty good at that, and it can form some biofilms sticking together. Here we can see, as I see this image with these pili, love is in the air, all right? We should have a parental advisory sticker right here, right? There is love happening between these two bacteria, all right? They are conjugating, all right? So the pili allows for genetic exchange. And like I said, typically it's plasmid stuff. Sometimes we might transfer some actual chromosome material along with that plasmid material, and then that creates a bacteria that's got a little bit extra going on, all right? We'll touch on some of that stuff a little bit later. But it can be two of the same types of bacteria, same species, but one's got the plasmid and the other doesn't. Or it could be two different species, all right? And that's how we can see things like drug resistance jumping ship, going from this type of bacteria that's got the resistance into somebody completely new and novel, and now it's got a similar type of resistance. That could be bad news, all right? Just depends on what they're sharing, all right? So here is an image of some of the flagella. Gram positives and negatives, again, rod-shaped bacteria, can have the opportunity for this type of locomotion. There are some similarities and there's a few differences. For our purposes in here, you don't need to get bogged down with any of this stuff, all right? This is super complex, all right? And really for what we're thinking about in here, you don't need to know all of this. This is basically just the motor for the flagella, all right? And in bacteria, I like to think of it more like a boat, like a propeller. And in eukaryotes, their flagella are different. It's more like a whip, all right? And they go back and forth like this, all right? Kind of snake-like, sort of. But with bacteria, they spin, all right? And it depends on what's up, all right? We think about them really like some smells, if you will. They don't have noses, but they can smell chemicals. And we call it chemotaxis, smelling some molecule, like maybe some sugars, right? Something like that, that may stimulate that bacteria. It's like, oh, something sweet over here. I'd like to go there, right? And it doesn't just turn and go right over there, but it gets there by a series of runs and tumbles, right? The flagella, when they are spinning counterclockwise, if it's one, it's just going. But if it's a bunch of flagella, they all function together and they spin counterclockwise and we move forward. And then we need to change directions because we don't just go, right? And they flip it into reverse and they turn clockwise. But when they turn clockwise, they don't all work together. They just do whatever the heck they want, and you tumble and you change directions, right? So it's like trying to turn around on the road, and instead of just making a nice smooth turn, you got to do like a multiple point turn, right? Runs and tumbles are really kind of bacteria making multiple point turns, but they do get where they need to go, all right? So I described this a second ago. Monotrichus, the single flagellum, Amphitrichus, two at either end, one at either end. Lopotrichus is a tuft, just a group of flagella all on one end, like a little bit of hair on the top of them, all right? And Paratrichus, they're all over, all right, completely covering that cell. And when they all spin counterclockwise, it's pretty impressive to see all of them just functioning as one big propeller, right? This one, well, that just makes sense. There's only one, all right? So the tumbling, change in directions, and the running, dependent upon which way we're turning our flagella, all right, as it spins around, all right? And as we see here, tethered into the inner membrane, going through the cell wall or through the outer membrane. You know, same idea though, as we get in here, basically serving as a motor to spin them around like that. So that's the idea with this chemotaxis. Like I said, it doesn't here. I mean, it looks like we should just turn and go right there, but we don't quite get there immediately, right? It takes a little more movement through there. We can stain up flagella. It's very difficult, but we can also inoculate some of these bacteria into some tubes that we call deeps. So it's got agar in the tube that's solidified straight up. Oftentimes we work with some slants where it's solidified on an angle, but those that are just filling that tube up with the agar, you use a different tool, a needle instead of a loop, and you just poke the bacteria straight down and pull it straight out and incubate it up. And if that bacteria is motile, it will grow out into the medium and turns it turbid. So you can see, sort of looks like an upside down Christmas tree as you have stabbed it in there, all right? And if it's not motile, you can see where you stabbed your needle in there, and the bacteria is just growing right along the needle stick, not spreading out into the media, all right? Kind of cool, all right? And you'll see a little bit of that as we move through the lab. So with the eukaryotes, there's a boatload of stuff going on inside of these cells, a lot of information, truly, when we look at this image. And I'm not going to go through and spout off all of this. We've got a few slides that will talk a little bit about it, all right? Considerably larger, about 10 times the shape or size of our bacterial cells. This one's got a flagellum on there, all right? They may or may not, all right? And depends on if we're talking, you know, animal or plant cell. Here's a single-celled eukaryote, a flagellate, all right? With its plasma membrane, no cell walls with these guys, right? And lots and lots of organelles on the inside, endomembrane system pushing plasma membrane to the inside and doing different jobs on it instead of just having it around the periphery, right? It's really kind of the idea with the endomembrane. So there's all sorts of different types of eukaryotic cells. that we think of as microbes, right? And in turn, some of them that we'll focus a little more on as we progress through, all right? These trypanosomes are a type of blood parasite. I think, you know, I don't want to see this in my blood smear, but when I see this, I'm like, oh, those are so beautiful. Little purple flagellates. I think they're awesome, all right? I also like this one down here. This is Plasmodium. This is the culprit that causes malaria. So inside of red cells or living beside red cells, two different types of parasites causing completely different types of infections. This paramecium right here is a ciliate. The outer bit of it are a bunch of little hair-like projections, makes it look sort of fuzzy, right? And I'm going to skip over Bordicella and our Chromelinia. So typically, look, I mean, this is an electron micrograph, but if you're looking at a eukaryote through the microscope, almost always the nucleus is going to be pretty apparent. You're going to recognize it. It will stick out, all right? If you're seeing red blood cells, you're not going to see a nucleus. They don't have it, all right? It looks different. But if you're looking at a blood smear, you can also tell that really pretty easily. Lots of red cells and then lots of white blood cells that do have nuclei, right? All over the place. So here with some fluorescence microscopy, we can see some of that with the nuclei stained up and then the membrane around it glowing in green. Part of the proteins making up some of the cytoskeleton in a darker green with that. So all sorts of membrane structure inside of these eukaryotes. This first bit showing the nucleus, a bilayer, but truly it's a double bilayer because part of the endoplasmic reticula, the rough endoplasmic reticula specifically, butts up right around the nucleus because lots of proteins are going to need to stay away from the cytoplasm. And we're going to process and produce them inside of the rough endoplasmic reticula, not free floating on ribosomes in the cytoplasm, all right? So more of the endoplasmic reticula, there are smooth endoplasmic reticula that lacks the ribosomes. Golgi apparatus is more of this endomembrane system, not really directly attached, but in close proximity. And we can see molecules packaged up, proteins or other things, in vesicles that bleb off of the rough endoplasmic reticula. Other substances coming off of the smooth reticula, they fuse to Golgi, they get collected, processed, packaged up, and exported out, right? In another vesicle, maybe becoming a lysosome full of digestive enzymes, or maybe going to the membrane of that cell, becoming receptors or other proteins studded on the surface. Just sort of depends, right? So Golgi truly processing and packaging things, shipping them through as we move along, all right? There are other substances that we might see like peroxisomes that allow organisms the ability to detoxify things, different lysosomes that also help with some of that. More of the proteins forming part of the cytoskeleton, the internal structures of our eukaryotic cells have a handful of different types of proteins that tether things in place, allow for the potential for some movement, whatever it might be, or aid in the process of division. And we can see stained up with some fluorescence microscopy, some of these different types. So with that, that also gives us some opportunities for these cells to move. Maybe a white blood cell or an amoeba, as we see the amoeboid movement, by pushing out their cytoplasm and pulling themselves through their environment on these pseudopods, right? There's other potentials for the movement of molecules inside of the cells and division. So there's multiple ways these cells may be connected and communicate, potentially walking or moving substances, which is pretty impressive, right? And we're truly, most all of this last little bit is not stuff that we're going to focus on, at least in this exam. We will talk some metabolism and details with mitochondria, but I'm not going to focus on that. Chloroplasts and the mitochondria, we think a little with the endosymbiotic theory. I think we showed some of that previously. And then the endocytosis is bulk movement of stuff, whether it be eating or drinking, things that can't just readily get into that cell through passive or active transport. We pull it in a membrane-bound vesicle, and exocytosis pushes it out, right? The removal of that stuff. All right, questions or concerns? Farewell, yes. Yes. Yep. Good day, good weekend, farewell.